Transfection. First, we start with a cell culture insert. In this, we have media and cells. The cells are what we are going to transfect. These are mammalian cells for this instance. First, we must start with the plasmid. The plasmid is a ring of protein or array of DNA that codes for a specific protein. In this case, green fluorescent protein. So first, we must pipette the plasmid in with the cells. Some of the cells will actually take the plasmid up, others won't. Those that do take out the plasmid will produce the protein.
And now for something completely different.
Hello, I wish to make a complaint. Hello, miss.
What do you mean, miss?
Oh, I'm sorry. I have a cold. I wish to make a complaint.
Sorry, we're closed for lunch.
Never mind that, my lad. I wish to make a complaint about these cells I purchased from this boutique not half an hour ago.
Oh, yes, the L99 mouse fibroblast cells. What's wrong with them?
I'll tell you what's wrong with them. They're dead. That's what's wrong with them.
No, they're just resting.
I know dead cells when I look at them, and these are dead cells. I tried transfecting them not 20 minutes ago, and they are untransfected.
Untransfected cells?
Yes.
No, they are transfected. Remarkable cells work very well with magnetofection.
No, they're untransfected.
No. See, you have to use magnetofection.
You see, magnetofection is a highly efficient transfection method that uses magnetic fields to guide particles containing plasmids into cells. Cationic magnetic nanoparticles are attached to the plasmids. After these cationic nanoparticles have been coated onto the DNA, they are concentrated in a solution and transfected into the cells, aided by an external magnetic field. And now for the math montage.
And that, my lad, is how magnetofection works.
All right, then. If that's the way I have to transfect them, then why aren't the genes being expressed? Did you take into account the osmotic pressure when you decided to use magnetofection?
That isn't...
No, you didn't. That's not osmotic pressure.
Yes, it is.
No, it isn't. See, this is how osmotic pressure, it releases the DNA, and we can model it thermodynamically.
Look, osmotic pressure is the pressure that water applies on the inside of the vacuole of a cell. As transfection occurs, the cell creates a semi-permeable vacuole to contain the plasmid. This vacuole allows water to flow in and stops the plasmid from flowing out. A difference in concentration is formed, and water then flows into that vacuole, causing it to eventually burst, releasing the plasmid into the cell. Now for the math montage of osmotic pressure.
Well, it's obvious then. So they did that now.
No, oh, really? Oh, really? Let's see. Hello, cells. Testing. Is this your 9 o'clock wake-up call? Well, I'm not sure that needs any further explanation. They're dead.
No, they're stunned.
Stunned?
Yeah. You stunned them, just as they were doing the transfection now. The L929 cells stun easily, Major.
Look, matey. I definitely have enough of this. Those cells are definitely not stunned. And you assured me not half an hour ago that these cells, their lack of movement was due to a prolonged night of multiplying.
Well, they're probably, that is...
What kind of talk is that? Look. Why did they make that farting sound when I opened it up? I should say that's not hardly normal.
The L929 fibroblast cells prefer keeping in a gas-enriched environment.
Look,
Remarkable cells like this, quite lovely flagella.
Look. I had the liberty of examining the cells when I got back to the lab. And I've discovered that the cells hadn't even expressed any protein.
Well, of course they hadn't produced protein. You didn't use magnetofection to get the DNA into the cells, nor promote osmotic swelling so that the genes were expressed.
Osmotic swelling? Mate, I'm the one that told you about the osmotic pressure and how it works, and it calls magnetofection. And you're telling me that the cells needed to do that in the first place? Now it sounds like you need to get your story straight. But mate, these cells are dead.
No, they're stunned.
It's not stunned. It's passed on. The cells are no more. They've ceased to be. It's expired and gone to meet their maker. These are late cells. The stiffs bereft of life, and may they rest in peace. If you hadn't glued them to the tube, they'd be pushing up daisies. The metabolic process of history. It's off the twig. They kicked the bucket. They shoved off the mortal coil and run down the curtain and joined the bleeding choir invisible. These are X cells.
Well, I'd better replace them then.
If you want to get anything done around here, you have to complain until you're blue in the mouth.
Sorry, Gov, fresh out of cells.
Oh, I see. I get the picture.
I've got a slug.
Slug?
Yeah.
Is it already transfected?
Not really.
Well, that's scarcely a replacement, is it?
No, I guess not.
Well, at least I understand how the magnetofection is used to get the genes into the cells. By subjecting the plasmid in the nanoparticle to a magnetic field, it can be easily driven into the cell for expression.
Yes, and then osmotic pressure is the whole thermodynamic mechanism by which the process works. As the osmotic pressure increases on the inside of the vacuole created by magnetofection, it then ruptures and releases the genes into the cell so that the protein can be produced.
Well, at least that got sorted out. You still have the slug?
No, I'm sorry, this is just getting too silly. Get on with it. Get on with it.
I'm sorry. Now, a frontal nudity.
Oh!